Okay, I'll give you a little introduction. Okay, let's get going, folks. So today we have with our instructor, Olena Vaske. Olena has had an interesting life history. Olena grew up in the Ukraine and came to Canada for her education. She got an undergraduate degree at the University of Toronto and then her PhD at the University of British Columbia. Then to Santa Cruz to join the Genomics Institute here as a postdoc for David Hausler. And I'm very interested in things that are clinically related and spent almost 2 years at UCSF with a clinical fellowship in genomic medicine. And having seen her at Santa Cruz, we brought her back as a faculty member. And Olena is now just newly tenured, so now she's a permanent faculty member. And so she's doing this fascinating project. Did you know that the genomic analysis is a big thing we've got going here at Santa Cruz? The world's genome browser is here. And so we have a real great core of scientists who can analyze these data. Olena has started something called Treehouse Project, which she will tell you about. That will be Olena's starting. Olena.
Thank you very much for that generous introduction, and I'm very excited to speak to you today, and thank you for inviting me. So this is pretty casual, and please stop me if something doesn't make sense or if you want to elaborate on something. So I'll tell you a little bit about pediatric cancers in general, and then the genomics of pediatric cancers, and specifically my interest is RNA genomics, so focusing on the RNA. And I think you've heard about RNA a little bit from the previous lectures, but I'll also give some introduction just in case. Okay, so I study pediatric cancers, the cancers in kids. And so unfortunately, kids do get cancer. Fortunately, it's more rare than in adults. So here are just the basic statistics. So in the US, we have about 42 children that get a cancer diagnosis every day. So in the whole of US. The average age of diagnosis is about 6 years of age. It is slightly more common in boys compared to girls. We don't know why, but it is slightly more common. So boys are 1 in 300, girls is 1 in 333, something like that. And it is a big problem. So in developed countries such as the US, this is the #1 cause of death by disease in children. So cancer is kind of the main disease killer of children. So when we talk about childhood cancer, we often say, you know, this is a medical success story. And I don't know, there is a documentary that's based on this book, Emperor of All Melodies. I don't know if some of you have read it on or seen on CBS. That kind of describes the evolution of cancer treatment. But so on this graph here, I'm showing the improvements in cancer treatment, in pediatric cancer, childhood cancer treatment that we've made. So in the 50s, virtually every child who received a cancer diagnosis would die. This was a lethal disease. In the 2000s, we now have about 80%, so most of the children will survive their diagnosis. On the y-axis, so you notice here I say 5-year survival rate, so 80% of children will survive for 5 years. This is the metric that's kind of been used for cancers in general, which is mostly adult cancers. And so if you think about it, for kids, so why I say, so this is, you know, a success story. So we went from 10% survival, so virtually no survival in the 50s, to 80% and now approaching 90% in the 2020s. But the metric that's used is 5-year survival rate. And so for kids, you know, you may argue 5-year survival rate, that's not, you know, you want to be, you want to cure their cancer, because if they're diagnosed at 6, they survive for 5 years, you know, they're still a child. The intent is different. So for adults, we may be okay with extending life, significantly extending life, but with kids, we really need to cure them so that they can go on and have 30, 40, well, more than that, ideally, years of life. And so that's why I say it's the beginning of a success story. Yes, we've made success, but we need to do more work. And so here is the problem. The long-term outcomes for children with cancer are unacceptable and can be improved, right? So if you now look at 30 years, so if you look at 5 years since diagnosis, 80 and now 90% of kids will survive for 5 years. But if you look at 30-year follow-ups, so 30 years since their diagnosis, and so they'll be in their 30s, right? So only 22% of children survive and don't have any chronic medical conditions from their treatment, only 22%. 34% will die within the 30 years of diagnosis. And then another fraction will survive, but will have, various types of chronic medical conditions as a result of treatment, including secondary cancer, including cardiac issues, neurological issues. And so we have work to do to improve this. We also have some diagnoses. So probably you haven't heard of this tumor. Hopefully you haven't heard of this tumor called diffuse intrinsic pontine glioma. This is mostly a pediatric, a childhood tumor, occurs in the brain. So this is a brain cancer. And this tumor actually killed Neil Armstrong's daughter, Karen Armstrong, in 1962. And so when we compare 1962 and today, in this disease in particular, this tumor remains terminal at diagnosis. So there is no effective treatment. We didn't have effective treatments in the 60s. We don't have effective treatments now. The only thing that's done is radiation to the brain, and it only works for a couple of months. And so when I talk to pediatric cancer families, they often say, well, we were able to put the man on the moon. Like, how come we still don't have cure for cancer? And so this is this is kind of part of why, yes, we've made success in treating childhood cancer, but we have a lot of challenges, diseases like this, and the long-term outcomes that I mentioned, which are because we treat very aggressively, we treat with very aggressive chemo and radiation at a very young age, and that then causes consequences later on. Okay, so what about genomics? So I mentioned that I study pediatric cancer genomics. So what is genomics? And you've probably seen this, so this might be repetition, but just in case. So all of our cells have DNA, right? And so DNA, genetic code, provides a set of instructions for how the cells function, how the whole body functions, for what we look like. So things like eye color, for example, are encoded in the DNA. Things like predisposition to various illnesses can be encoded in the DNA, or traits like height, weight, and partially encoded in the DNA. And so the analogy is our DNA provides a set of instructions, kind of like a cookbook, right? It has many recipes for how the cell should do certain things. And so that's the DNA. And then the outcome, so then from a cookbook, you can follow a recipe and make a meal. And so other molecules like RNA and protein will be involved in this and actually implementing kind of the instructions into something that is relevant. And so genomics then is the study of DNA, of the whole genome, and also the relationship between the instructions and the outcome, how these instructions then encode something relevant. So cancer is a genetic disease, okay? And nowadays we actually say it's a genomic disease. And the difference between genetics and genomics is kind of larger scale. So genetics will only look at small pieces of DNA. Genomics says, okay, let's look at the whole genome, at all of the DNA together. And so cancer fundamentally is a genetic disease. It's a disease of genomes. So we have here normal DNA, which has sequence of letters. So the DNA code has only 4 letters, A, T, C, G, and then provides a set of instructions for how the cell should function. And you've heard about this in earlier lectures. And so in cancer, we have errors. Basically, cancer happens when we have errors or spelling mistakes, if you will, in the DNA that change some of these letters, and now you have abnormal instructions, and so the cells will behave abnormally, and namely they'll proliferate, they'll grow uncontrollably and become cancerous. And so fundamentally, cancer, we say, is a genetic disease. Here is kind of an outline. So this is this is normal skin, and it has nice epithelial, we call them epithelial skin cells that are lined up. And then here in the middle, I have a cluster of blue cells. And so these cells presumably accumulated some spelling mistakes in their DNA that make that these mistakes, or mutations we call them, now make them grow uncontrollably and become cancerous. So they divide more rapidly than normal cells. They divide inappropriately, they grow. Eventually, they will be able to move around, or we say metastasize, so leave this side and go to another side in the body. And so that's cancer. So where do we get mutations? So why do we get mutations in the DNA? Well, actually, mutations accumulate over our whole life, starting from when we were a single cell, and even before that. So with every time DNA replicates, there is opportunity for a mutation to happen just randomly. And yes, we have repair mechanisms. So we have mechanisms in the cells that you may have heard about that will try to repair spelling mistakes, or proofread, if you will, but they're not perfect. So nothing is perfect. And over time, just by virtue of living, we accumulate mutations. So from the moment that we're a single cell throughout our life. So due to intrinsic processes, and then also once we're born, we're exposed to environment, right? And as we grow up, we have some diet, smoking, we know, is a big carcinogen, right? So there are environmental or lifestyle processes that could contribute to mutations, and some of them we know about, others we don't know about. And so as a result of these two processes, or multiple processes, both intrinsic and extrinsic, we accumulate mutations. Any questions so far? And so here is a slide that kind of highlights the point. That cancer is an evolutionary process. So cancer doesn't just take one spelling mistake. It takes several spelling mistakes to happen. And you can think of the cancer as an evolutionary, as evolving. I'm sorry. And so with acquiring more and more spelling mistakes, the cell becomes kind of more and more cancerous. So initially it can proliferate in kind of its place, and eventually it acquires enough mutations to metastasize or move around, so leave its original site and go and colonize kind of other parts of the body, and we call that process metastasis. And so all of that is all of that happens because of accumulation of additional spelling mistakes. I'm sorry. So here I wanted to just point out that not all mutations contribute to cancer. And so, we have mutations that occur. That's fine.
Don't worry.
I don't have COVID. I had COVID a few weeks ago, so I'm wearing out just in case. But I'm COVID negative. Sorry. Okay. Let me take a second.
Yeah. Would it be better without?
Maybe the hips, if you take the mask off, I don't mind.
Well, I want to be careful. I don't want to give you what.
No. Most of us wear masks. Wear masks.
Sorry. Okay, I think I'm fine. Okay, maybe I'll speak about last one.
That's great.
Okay, thank you. So we have driver mutations, and so these are mutations that actually give a cell an advantage, like a growth advantage, make it metastasize, give it some kind of property that favors that cell over a normal cell. And then passenger mutations are just there for the right, so they don't do anything. And this is why this is relevant. It's just, I think, interesting that not all mutations that happen throughout our life are going to be cancerous.
Is it known that, the characteristics of the driver was the passenger?
So this is
Are
the
there
big,
some attributes for each that would have?
yeah, so that's a great question. And so this is the big kind of question in genomics, how to distinguish the two. Because, so I'm going to say later that we now have the technology to read cancer genomes, kind of in the last couple of decades. We now have the opportunity to read cancer genomes and identify mutations. And so the first thing we realized is not all mutations that we see mean something.
Mm.
And so this is a scientific question. How do you tell if a mutation gives the cell some advantage or is it neutral? And so I just included this slide just in case in the previous lectures you talked about some of the kinases and cell cycle growth factors. So a lot of the driver mutations that we know about actually target these type of functional categories. So these are genes that are involved in the control of cell cycle, involved in signaling. So they have some very important function normally in the cell. And so then in cancer, these genes will mutate. And so this function is now abnormal, like either for cell cycle proteins, the mutations in these genes make the cell go into cell cycle and divide, right, uncontrollably. And then there is two types of cancer driver genes. There's oncogenes and tumor suppressors. So you may have heard of these or maybe not. But so oncogenes are genes, when they're activated or when their function is enhanced, that's bad. That makes the cell more cancerous. And tumor suppressors are kind of like gatekeepers. So they're the opposite. They're you could think of them as policemen. So they normally go around and check that everything's okay, everybody is behaving, the cell's behaving normally. But then if you lose that function, there is no control, then the cell can behave more abnormally. So there are gatekeepers, and so losing tumor suppressor is bad. So gaining oncogene function is bad, and losing tumor suppressor function is bad. So there's kind of two categories of genes, which is interesting. One kind of interesting piece of information, and I'm going to later talk about targeted therapies, how this is important for cancer treatment. So treating oncogenes or having a therapy that will inactivate an oncogene is what we can do, because it's easier to inhibit a function than to restore. So for tumor suppressor, you kind of need to give that function back, and it's much harder to do. So most of the cancer therapies we have target the oncogenes. I also wanted to highlight this issue that makes, why is cancer difficult to treat? And in the case, I've mentioned this diffuse intrinsic pontine glioma, the pediatric cancer that's lethal today, that killed Neil Armstrong's daughter. And so one issue is tumor heterogeneity. So what this means, if you take a tumor, which is a collection of cancer cells, a collection of cells that have, some mutations, not every cell within that tumor is the same. So cells can have, it's like a community. You can think of it as a community. Each member of the community is unique, may have different mutations. And so we call that process we call this phenomenon tumor heterogeneity. And so it's very, very hard then to get rid of the whole tumor, because every cell is kind of slightly different. And so you may need to treat like with many different things to kill every single cell, but then it becomes toxic for the patient. You can't, give 10 different drugs at the same time. So this is
Sort of all those variations from the founder cell.
So that's a really great question. And so, this is highlighted in this figure. So presumably, in the beginning, you have normal cells, and then one normal cell acquires a spelling mistake. So it's the founder cell. and maybe the cell grows slightly faster, so it has an evolutionary advantage. And then its daughter cells, here shown in red, also will have this spelling mistake. But then in this population of red cells, there is another cell that acquires the second spelling mistake that gives it, some other additional advantage. And so then daughters of this purple cell will have 2 mutations, but then you'll still have some red cells. So it's, yes, kind of descendants of each cell will preserve kind of the founder mutation, but then there'll be additional mutations that are acquired. So you can really think of this as a community. If you have heard about like animal species, right? Like animals will have different variations in the population. Cells are the same, or cancer it's a community of cells that each has different features, and that's why it's hard to kill the whole community. Right, and it's especially hard when you diagnose cancer late, right? So when you diagnose cancer late, you already have many different populations there that you have to target. If you catch it early, like at this stage, you only have one cell, or here when you have a couple of populations, it's easier to target. But when you're late, you already have many. You have a lot of, we call heterogeneity, that you need to target, and maybe at this time already spread to different organs, right? So that's what makes it very difficult.
Some cancer cells grow faster than others. Right?
Absolutely, yeah.
What's the difference? What is causing some cancer cells?
Great question. So
The environment, the outer environment, or is it the cell itself?
So it's both. So that's a great question. So in the cancer, some cells may grow faster than others. You also know, you treat a cancer, and often some cells will die, but then others will not die, right? And then regrow. And so that's due to differences in mutations. So some mutations, maybe cells that grow faster, they have some additional mutation that makes them grow faster. A microenvironment or environment also matters, because maybe they have a mutation that make them survive better in this particular environment, like in the lung, for example. So it's kind of both. It's mutations.
Prostate cancer, I understand, goes very slowly.
Yeah.
Is it prostate cancer?
Yeah, prostate cancer.
That must be the outer environment which causes the cancer slowing down.
Yeah, so prostate cancer is a hormone dependent, or it starts often, right, as it's fueled by hormones.
Yeah.
Similarly, ovarian and breast cancer in females as well. And then it evolves to be kind of hormone independent through this evolution. So absolutely. Yeah, but initially it selects, like the environment selects for cells that thrive in that environment. So it is kind of like evolution of species, but at the cell level. Mm-hmm. This concept is why it's difficult to treat cancer. Also, if you consider different patients, because or different people, we're all genetically different. We know we have differences. We look differently. We have different backgrounds. So we have a lot of differences in our DNA. And so the cancers that we all have are different, like by virtue that the starting cells are different. And so really it's not one disease, but as many diseases as there are people, right? And so that's, you can of course find commonalities between different cancers, but no 2 cancers are exactly the same, which makes it also very difficult. So that's why cancer is a difficult beast to beat. Okay, so what is the promise of genomics? So genomics as a discipline kind of emerged in the past few decades. And it's really been fueled by technological advances. We call it DNA sequencing, which is a technology that really allows one to read cancer DNA. So read these letters and identify potentially spelling mistakes, identifying mutations. And so as Mary mentioned, UCSC has a history in genomics and a great legacy. David Hausler and Jim Kant were involved in kind of the initial human genome project and developed a computer program called the UCSC Genome Browser that was able to put the sequence that's being read on the website so everybody can see it, right? And so we can look at it and we can, look at this genome and the human genome at the different areas. Everybody in the world, including high school students or people from every walk. This is a free program. If you're curious, you can go and view, you can type in a gene name and view a region in the genome. And so since then, kind of UCSC has been very active in the area of genomics. But when so the first human genome was very expensive. It costs like multiple billions of dollars to sequence one human, right? And we were very excited when that was done in the early 2000s, or it was partially done. We only fully completed it a few years, like with a telomere to telomere. But the first kind of draft sequence early 2000, we were very excited, but then we've realized people are all very different. So it's not enough to just read one genome because it doesn't tell us about every one of us. So we really need to read everybody's genome and understand what are the areas that are different between people. But now we have, so since that first human genome, there was a question, I'm sorry.
Yeah.
Yeah.
To identify a mutation, you have to know what's correct, right? And so for the human genome, I've always been perplexed. You actually map all the different allele combinations that are available. So you know everything that's correct, you would have to be able to identify each individual.
Excellent question. So, when I say spelling mistake, well, what's the correct sequence? And so that's why, so the first human genome, and actually there were two kind of first human genome projects. I don't know if there was a public human genome effort, right, funded by public money, which UCSC was a part of. And then there was a private company called Celera that was trying to also do that at the same time. And so then, there was negotiation. And so the announcement, they kind of finished together, right? And so we have two kind of versions of the genome. We have the Celera private company and then the public version. And so initially what we said, okay, we call that reference sequence and we say that is correct, like whatever. But then we've realized it's not a full story because all of us have differences. And, different ethnic groups have differences in the sequence, but also, just different people in general. And so we really need to catalog all the different alleles, like all the different versions of the genome. And that's a big effort right now. So actually UCSC is involved in that. It's called the Pan-Genome Project to create a comprehensive normal reference. So we're not done with that yet because actually, human genome is big. I think I heard an analogy, if you line up all the letters, you can travel from the Earth to the moon and back. Like that's how many letters we have in one copy of the genome, and we have two copies, one from mom, one from dad. And we can have mutations affecting only one copy or affecting both. So there is a lot of complexity. And so it's not easy, but we're really, as a field, trying to think about that because you need to really understand what's normal before to interpret this variance. What we do in cancer. So in cancer, what we also have is, so cancer affects some organs, right? So usually, you have a tumor in the breast or in the prostate, but then the rest of the body is okay. And so what this means is that those spelling mistakes only affect, say, the prostate cells. They're only found in the prostate or the breast or wherever the cancer is. And so what you can do in the cancer is you can actually compare unaffected tissue or the DNA from unaffected tissue to affected tissue to understand what spelling mistakes exist in the prostate compared to, blood or some other tissue. And so that's good news. So in the field of cancer, we're able to interpret these spelling mistakes by comparing, we call tumor to normal tissue in the same patient, because then we know what's the reference in that patient. In germline genetic diseases, because of course, there's also diseases like birth defects, we call them germline, which means every cell in the body, like DNA in every cell in the body is affected. That's much harder. And there we really need to know all the allelic variations to know what's normal and what's abnormal. But for cancer, we can utilize the sequence in the normal unaffected tissue. And that's what we do in the field. But that's an excellent question.
So when you identify those mutations, they're somewhat specific. Does that inform the treatment, the specific treatment, or is it just like one size fits all when it comes to?
So thank you for that question. So I will, yes, so I'll get to that. So once with the advent of genomics, kind of came with this promise, right? So when I was in, and I've kind of I'm not, well, I'm in my like late 30s, but I have seen this develop in my lifetime. So when I was in my PhD, genomics was coming of age, and we were so excited because we thought, okay, we're gonna read cancer genomes, understand the mutations, and then treat everybody. Like we're gonna cure, like that's what I thought going into PhD. We're gonna cure cancer in my life, in my by doing exactly that, by identifying, okay, this patient has this mutation, we're gonna treat with this drug to kill their cancer. But it turned out to be more complicated than that. One issue is heterogeneity. So one patient doesn't just have one mutation. There is, hundreds sometimes of mutations, and how do you target all of them? Do you need to target all of them? So there's more complications, but that was the promise, is we're going to read everybody's cancer DNA, find the mutation, treat them. What I want to get back to is pediatric cancers. So I've mentioned to you that cancer is caused by mutations, right? And there is an evolution. So as a cancer develops, there is also more mutations in the cancer. If you compare early cancer to late cancer, late stage cancer will have more mutations. Pediatric cancers have, so this is a complicated graph. I apologize, but this is a real graph. On the x-axis, we have different types of cancer, and this is pediatric. These are all different adult cancers. On the y-axis, we have mutation rates. So every one circle is one cancer patient. So these are all patients here that have a childhood cancer. And, this patient has 0.2, some units of mutations. This patient has more. But if you line them up, so what I want to tell you from this figure is pediatric cancers on average have fewer mutations than adult cancers. And that makes sense probably because I said mutations accumulate over lifetime. So the earlier you are, the fewer mutations you have because you didn't have that much time to accumulate mutations. You maybe were not exposed, kids like don't smoke, you were not exposed to mutagens, and so you have fewer mutations, but you still get cancer. And so fundamentally, there is something different about the biology of cancers that happen early in life compared to cancers that happen late in life. And these are just examples of adult cancers, pediatric cancers. So on average, of course, there's a lot of variation, a lot of complexity, but on average, cancers that happen earlier have fewer mutations.
Does that mean that they're easier to treat?
Very good question.
Can you repeat the question?
Sorry, the question is, does this mean that cancers that happen in younger people are easier to treat? You would think, but not necessarily. The survival overall, so when I showed you this graph, right, then I said, say 80% and now 90% survive. This is better statistics. Like if we look at all adults, like just all adult cancers, the statistics are not like this. It's much worse. But this is because kids can withstand chemotherapy much better than, somebody who is elderly maybe and has some comorbidities, some other things, right? So it's not because they're easier to treat, it's just because the aggressive treatments that we now use for kids, you really can, the doses that we give to kids are just unimaginable. Adults would not survive that dose. So that's why it's not that they're easier to treat. But you would think, they're easier to study, and that's why when I talk to my colleagues, I say you should study pediatric cancer because they're easier model because there is fewer noise, but there is less noise, there is fewer mutations, and by understanding pediatric cancer, you can then understand adults. So because when I mentioned not all mutations are cancer causing, right? There's drivers and passengers, so adults have a lot of passengers just by virtue of having more mutations. If you sequence like a lung cancer, it can have thousands of spelling mistakes. And then you have to use computer programs, and this is the field called bioinformatics, to sift through all of those spelling mistakes, understand what is, what is a driver, like what mutations are relevant and what are not. If you sequence, and by sequence, I mean read the DNA, a pediatric patient, in an extreme example, cancer called retinoblastoma, which is an eye cancer, only one. Like some retinoblastomas have only one mutation. So the genomes, we call them quieter. They're much easier to understand. And, what you learn in kids can then be relevant to adults because it can help you identify what are the driver genes. But anyway, so yeah, it doesn't, so this is the paradigm, kind of what you were talking about. Okay, what is the promise for treatment? So yeah, we have this cancer genomics, cancer is due to mutations. We're now able to read the DNA, identify mutations. And so here is the paradigm. So, you know, and at the beginning of genomics, this is what we were hoping for all cancers. You know, you start with a heterogeneous patient cohort, you know, and I should back up. So before genomics, cancer was kind of subtype based on which organ it was in, right? So, you know, you go to a doctor, the cancer is in the lung, they say, okay, it's lung cancer. Lung cancers we treat like this. So it was like one size fit all. You know, then later we started to look at the cancer under the microscope and kind of see, okay, it's lung cancer, but the cells look like adenocarcinoma versus squamous cell carcinoma. So, you know, we can, under the microscope, to group lung cancers into different types based on what it looks like. This is an extra resolution because now we can sequence the lung cancers and say, okay, you know, this lung cancer has these particular mutations, this other lung cancer has different mutations. And so we can then stratify patients and treat based on the specific mutations, kind of what you were alluding to. So that was the hope with this. And it is happening. So I shouldn't, you know, say for some tumor types, this has led to really dramatic improvements. For example, lung cancer, we've identified through sequencing that there is a subtype of cancer that has a mutation in a gene called ALK kinase. And so that subtype of lung cancer responds very well to therapy. And before we did the sequencing, we didn't know. Now we can identify a subset of lung cancers that really benefit from, you know, this therapy called crizotinib, and they have a much better outcome when on this treatment. But what I'm trying to say, we haven't solved globally, right? But then there are other examples where we don't have a good therapy. So for some tumor types, this has been hugely successful for adults, is being able to read the DNA, identify specific mutations, and then treat the patient based on that mutation.
Do they not also take the tissue, grow it, and just with, is it not a genomic question, but just run it against different drugs?
Yes.
Can that correlate with the, can you live for that?
Absolutely. So the question is, can they also, you know, without worrying about genomics, just take somebody's tumor, grow it in a dish, and test with different drugs and see which drug kills the cancer? And so that's absolutely being done. It's done in the research setting. So it's not routinely done for everybody in the clinic, but there are research programs doing that, including we are doing something like that. What we found is oftentimes the dish does not correlate with it's more complicated than it would seem. But it's absolutely being done, and people think about this. There are logistical complications too. So not every cancer you can grow successfully in a dish, unfortunately. Some cancers you can grow very well. For other patients, you cannot, you know, either they don't have enough cancer that could be safely obtained, or, you know, or it's not good enough. Like maybe it's not, other times it's very aggressive, but it doesn't survive in the dish for some reason. So there is challenge. So you cannot do it successfully necessarily for every case. B, it doesn't always correlate with outcome in the patient. Another limitation, it takes time. So oftentimes, so these type of experimental approaches are done, and this is something we need to change in the field, but this is done on a late stage cancer because this is still considered new treatment. So this is done when somebody has gone through chemo and like nothing else worked, and now they're looking at experimental therapies. And so this is done, but then by the time it takes to grow up the cell and test the drug, the patient is maybe sick, and which is something that's really disheartening. But it is being done in research setting. There are companies doing that. So there are companies that one could pay to have this done, and so some people do that. But that's definitely, I think we should do all of that. Like we should do that, and in combination with genomics. Yeah, so it's still a new kind of concept. Yeah.
Surviving a cancer cell in the dish, that reminds me. Is there something you can do with changing the environment and killing the cancer cells by increasing the temperature, increasing the body temperature, cooling it down, putting a lot of alcohol in the body or whatever. It reminds me of a doctor in the 60s in Germany. He had an institute in Bavaria, and he was actually increasing the temperature of the body, and he had some success with cancer patients. And I think it probably happened because there were certain types of cancer responding to that treatment and others not.
Absolutely, that's what I think too. I mean, there's a lot of stories, you hear some cancer patient tried some alternative approach and, then they swear by it and they say, and I think everybody's cancer is different. Like maybe that unique like, I don't know, because I can't say that it doesn't work. I also think that these types of approaches should be studied and so that we can document exactly what's happening, ideally in a study settings, so we can watch what happened and learn from it. And so if indeed it works, we can use that.
But you never heard anything again, increasing the body temperature, that's one way to kill cancer cells.
I mean, definitely in laboratory that works. I think, oftentimes like maybe you need to increase it too much. Like, so I'm not familiar with that particular investigator. I know it works in the lab. I'm just thinking therapeutically. So it's much easier to treat early stage cancer. So if there's only a few cells, like definitely. If it's a late stage cancer when the cells are all throughout the body, then it's much harder, and they're different. They have different mutations. That's much harder. Oftentimes those type of things, including hypoxia. So hypoxia, lack of oxygen, also kills cancer cells. But achieving that, like achieving a therapeutic effect, where you don't have enough oxygen that the cells will die, like maybe is hard. So I'm saying maybe the temperature that you need to achieve is not survivable. I don't know. So you might kill so I think that's the challenge, is we need to kill the cancer without killing the patient.
Yeah.
And patients may have comorbidities, like there is all kinds of considerations that I see there is a question maybe in the chat.
No, I don't think so.
I think this is, I think this is great discussion, but so what I wanted to say, so this is the paradigm of genomics. So using genomics to stratify cancers and then treat based on mutations. And so what I wanted to point out, that this paradigm does not work for kids. So for kids, what happens is this, yes, we start with a heterogeneous population of patients with the same diagnosis, like say neuroblastoma or diffuse intrinsic pontine glioma. We do genomics and we only find a mutation that could be targeted by therapy in like 10%. I mean, there is different statistics and depends on the tumor type, but it's the minority. If in adult, we could find potential mutations in most cases, in kids, we can only find it in some very few cases. And that's because we have fewer mutations in kids. And also some of the mutations are different. We call them undruggable. So we don't have currently drugs that target those mutations. And that's because pediatric cancers are rare. So the way that our pharma works, right, like if this target only happens in a few patients, in the country, it takes millions of dollars or billions of dollars to take something to market, then they wouldn't pursue. So there is a whole big other thing of drug development in rare diseases, right? But anyway, so only a minority of patients will benefit from this. And so in my research, what we hypothesized, we said, well, can you use RNA? So this is looking at the DNA, so looking for spelling mistakes in the DNA. But can you use RNA to identify additional targets for therapy? And so what does this mean? So we use the technology called RNA sequencing. So this is also sequencing, also reading, but not the DNA, but the next molecule up, RNA. Let me ask you, are you familiar with RNA from previous? Great, okay, that's amazing.
Right, they better all put up their hand.
And so RNA is amazing, in my opinion, because you can look, so you can still find spelling mistakes because you have the sequence, right, in the RNA. But you also can quantify gene expression. So here is a cartoon. So when we do sequencing, we kind of, we call them reads. So here is an example. So here is the reference sequence. So this is the sequence we think is correct. So to your question, sorry, I don't know people's names, but you know, the reference sequence, something we think is correct. And then you sequence, or you read a patient's DNA or RNA, and you obtain these reads, which are, you know, kind of letter fragments. So they're short because that's how the technology works. So you can't just read the whole 3 billion, you know, base pairs in one long word. What you do is you read like 20, well, not 20, now 100 letters here, 100 letters there. And then you have to computationally map it to where those letters came from. But then you end up with this. So you have some reads informing you about what's happening in this area of the genome. And so you can do this with the RNA too. And so you can spot, so SNP, single nucleotide polymorphism, is like a type of spelling mistake, if you will. So there's a difference between the reference and what you see in the patient. And so you can see that from the RNA. So you don't lose the mutation information from looking at the RNA. But what you also gain is you gain gene expression. So you can kind of estimate how much of a specific gene product maybe is made. If this gene is turned on or turned off in a particular cancer, and how much is it turned on, how much turned off, we call this gene expression. And so you've talked about that. And so you can get that from RNA sequencing, which is amazing. And this is a cartoon. So we have a gene here. In black, we have these reads that are mapping to exons, right? So you probably talked about splicing, right? How in the gene you have exons and introns. And so you can then quantify different exons, how you can even infer splicing isoforms from looking at the RNA. But, you know, RNA analysis is much more challenging. And specifically, so in my group, we look at expression. So, you know, yes, we look at mutations, but this is not a new thing. Everybody kind of, you know, knows mutations are important, and everybody looks at cancer mutations. What we're saying, okay, let's also look at abundance of genes. And that is challenging because, again, how do you know? So say if you wanted to find genes that are turned on too much in a cancer, what's too much? So to your point, like you need to compare it to something to say, okay, normally the gene should not be on, but now it's on in this cancer, and that's abnormal. So you need to know what the normal is. I mean, you know, you could think, okay, well, if you have a liver cancer, you can compare it to normal liver cell maybe, and then see, okay, what's turned on in cancer compared to normal liver. But that requires, you know, that patient donating normal liver, right? So in the clinic, we don't typically, especially for brain cancer, like a surgeon will not just take somebody's normal brain so we can have a comparison in the lab, right? So it's not practical. For childhood cancers, we often don't know what the true normal is because we think that some of these cancers happen very early in development. And so that cell type does not exist in a mature person, mature human. But anyway, so that's the challenge. And so what we in our group develop is this bioinformatics, so this computational method to compare a single patient to, we call it compendium, thousands of other cancer patients, and see. And the hypothesis is that, well, if this patient's tumor is different, you know, this gene is highly expressed in this patient compared to thousands of other cancers, maybe this gene is important because it's just highly on. And that's the kind of big picture idea. And so this is kind of the analogy, right? So to know how big this dog is, you know, you have no way of saying, I mean, unless you know this cartoon, how big this dog is. So you know by comparing, okay, so yeah, this is a big dog because the other dogs. And so this is the analogy we use for gene expression. We say, okay, we need to compare to other different cancers and say, okay, this gene is really highly expressed in this patient. And so this is what we do. And so, okay, so let me tell you, and how am I doing the time?
Well, so it's you have 40 minutes.
Okay, I actually don't have that much left. And I really enjoyed the discussion. So I think this is great. I have a couple of kind of patient examples of how that illustrate this approach. And this is the patient. So I'll tell you this about this patient that started our whole treehouse program. And I'll tell you finally what treehouse is. But this is the patient that really motivated this work. And so this was an 8-year-old boy with a very rare tumor. called an undifferentiated CNS, that's central nervous system, so that's the brain sarcoma. So normally sarcomas don't happen in the brain, but in this case, this was a tumor in the brain. This was a Canadian patient. So as Barry mentioned, I was educated in Canada, and so I have a lot of colleagues still in Vancouver and in Toronto. This patient came from British Columbia Children's Hospital, and so was treated very aggressively, as many cancers are, right, with aggressive chemotherapy, in this case, stem cell transplant, local radiation, because the idea is to kill the cancer and prevent it from coming back, because if it comes back, if some cells survive this aggressive treatment, then they're usually mutated, that they're usually very different from the original cancer, and they're much more aggressive. So treated very aggressively for 2 years, it was fine, you know, there was no cancer. But then 2 years later, the cancer comes back in the lungs, so metastasized from the brain to the lungs. And then at that point, there was no standard treatment options for this child. And so he was involved in a clinical trial in Vancouver called Personalized Oncogenomics Trial. And so it's this idea, so personalized means, you know, we find something that suits you, not one thing fits all, but, you know, this is really, this term is now kind of common with genomic trials, because the idea is that let's analyze this particular tumor and find something that fits you. And so in this trial, then tumor DNA and RNA of this patient was analyzed to find spelling mistakes that are specific to this patient. But as you see here, and this is kind of a common thing for these trials, this is done late, right? So already the tumor we know is super aggressive. So it's very hard to treat with anything at this point. And so I think what we want, and what is kind of the movement in the field, is to do these things earlier, right? Like, why don't we do this up front and see if there is therapies? But this is, you know, it took us years to get this there. But in any case, so this is what was done. So this DNA and RNA sequencing revealed one mutation in this cancer called a gene fusion. So don't worry about this gene name, but basically 2 genes were fused into one. And this is called the gene fusion, and these happen in cancer. We know about them. They happen a lot in leukemia cancers and blood cancers. They also happen in sarcoma cancers. But the problem is, so this is one of the pediatric or childhood mutations, don't happen in adults. Very often there is no treatment, there is no therapy to target that particular. We know this mutation is important, it's a driver, but we don't have a molecule to target it. So no therapies. And so then they shared, so these investigators shared RNA data with us at UC Santa Cruz. And so we've tried our approach, you know, this Clifford approach, where we said, okay, let's compare this patient to other cancer patients and see what's unique about this patient. So we thought we would try that. This is a complicated slide. I don't know if in previous lectures you saw kind of signaling pathways.
No, we didn't do that.
Okay, I think what I want to, okay, let me show you this. So we found, so JAK1 is a particular oncogene. So remember, oncogene is a cancer gene that when it's bad to have it overact. So this is not the gatekeeper gene, it's the other type where cancers want to have it super active, either through mutation or through something else. They want to turn it on abnormally because this gene makes signals survive, cells survive and proliferate and divide. And so in this patient, so here I have expression level of JAK1. So the amount of RNA that's made from JAK1 gene. Here on the y-axis, I have the amount, so we call it expression. And then we have different, so we have patient 1, our patient, then we have all cancers at the time that we had in our hands. And then we looked at sarcoma cancer, the lung cancers, because this was a lung metastasis. So this is the best we could do, is compare to cancers in the lung.
Was it already suspected that the JAK1 was involved?
No, it was not. So yeah, it was not suspected that JAK1 was involved. We looked at, so why did we look at JAK1 specifically? Several things. So we looked at things that could be targeted by drugs, where we know for some other indication, there is an FDA-approved therapy that can target JAK1, but maybe not in sarcoma, in some other disease, because there are databases of basically FDA-approved therapies and genes that they target. So that's the reason we kind of, because we were practically thinking, okay, what can we do?
But JAK1 is a mutated gene.
So it is mutated in other cancers. So it could be mutated in cancer. So it is a known oncogene. So we knew this is a cancer driver gene. We just didn't know this to be a driver in sarcoma, like in this particular type of cancer, but it is a driver in leukemia. So in blood cancers, this is often a driver. But so we noticed that this JAK was highly expressed in this patient. And so we hypothesized, this is a guess. We said, well, maybe JAK1 has something to do with why this cancer is growing, right? Like maybe this is a driver gene. And so this is a complicated slide. But what I want to take away, so there is an inhibitor called ruxolitinib, which is used in leukemia. So it's used in blood cancer. There is clinical experience with this drug because we know JAK is important for leukemias. One other thing that this is a pill, so it's not an IV drug. You can take it as a pill, so it doesn't have a lot of side effects. And so long story short, so the clinician decided to try this because there was nothing else to do. And they talked to the family and it's a pill, you know, what do we have to lose? And so they tried ruxolitinib in this patient. So this is kind of, so I'll summarize this graph. Basically, they administered ruxolitinib, the patient improved. They improved for some time. So he started playing, his weight improved. Then they noticed tumor progression. So at that time, the patient had multiple sites of the tumor. And so one of the sites started growing again. And so then they stopped this therapy because it's experimental therapy. And then the patient got much worse. And then actually the family said, well, he was doing very well on ruxolitinib, let's restart it. And so then they restarted ruxolitinib at the parents' request and then saw again improvement. Eventually, unfortunately, the patient passed away from this therapy. But for us at UC Santa Cruz, so I was in my postdoc at the time and I was doing bioinformatics, so computational analysis. And we were, you know, a team of people here at UC Santa Cruz. We don't have a medical school here. And we just did this analysis and some clinician acted based on this and there was success. And so we became very inspired and we said, well, can we ask the question systematically? If we look at other pediatric cancer patients, can we always find these kind of JAK1s, like these cancer driver genes that are very highly expressed and there's a drug? And can we test that? That could be a different therapeutic option. And so the patient's name was Kelvin. And so he, at that time was already older. So he was 11 or 12. And he knew that he was participating in this study and he was very proud of this. And so his family wanted us to use his name because this is an uncommon thing. You know, we kind of de-identify patients. We don't know the names. But in this case, the family wanted us to know the name and use his name in talks, which is what I'm doing. But we started Treehouse because of Kelvin. And we wanted to see if this kind of comparisons, so to big data, or we say, you know, collections of other cancer will help us identify novel treatment targets for difficult cancers. And so that's the hypothesis, scientific hypothesis, is that comparing RNA-seq data from single tumors to cohorts of cancers will help us identify these unusually expressed oncogenes that could be targeted by therapies. And so we started Treehouse Childhood Cancer Initiative with this. Initially, this was a partnership, UC Santa Cruz and 3 hospitals, so UCSF, Benioff Hospital, and specifically the brain tumor arm there, Stanford, Lucille Packard Children's Hospital, and Children's Hospital of Orange County. And so those are places that had patients. And then later we got some more partners, Nationwide Children's Hospital, Canadian partners. And so we were evaluating this idea. So taking RNA sequencing data from tumors, doing these comparative analysis that we developed, identifying drug targets, and then communicating to the teams, you know, providing this information. Of course, we're not doctors, so we can't say, oh, you have to treat with this drug, but what we have been doing is providing information and then the clinical team can decide what to do, right? So a lot of, you know, it's up to them. We just want to tell them what we found from this analysis. So we do a lot of data comparisons, and so we build these compendia. This is very important for us. It's kind of this contextual analysis, comparing one patient to all the other tumors, and the more the better. And so we build these compendia. This is a viewer that was developed at UC Santa Cruz by George Stewart's group, which we call it a tumor map. This actually uses Google Map software. So here every dot, so every small dot, it's a bird's eye view, but every small dot is a cancer patient or RNA profile of a cancer patient. And then the dots are laid out according to how similar different tumors are in the RNA space. And then they're colored by type of cancer. And so we have adult and pediatric, all kinds of cancers here. And so, for example, you see breast cancers, they form this kind of shape. You know, they are mostly similar to each other, but you can see there is different groups of breast cancer, which is kind of very interesting. What's very interesting to us is you have, you know, for example, this is a well, I'll show you this. You see this orange cancer, which is a osteosarcoma, but then you have green infiltration, which green is a liver cancer. So what it's saying, actually, this is a liver cancer, but it's more similar to another type of cancer, which is very interesting. And we didn't know before we looked at the RNA. But so now we have over 12,000 tumors, and we constantly grow this because we think the more data we have, the more we can learn. And we kind of do two things with this map. So we can first say, okay, this cancer, what is it similar to? So give me tumors like me. What are the most similar tumors from the compendium? And then can we learn from that? What did that patient respond to? You know, the similar. And we can also ask what's unique about my tumor. And we think that what's unique, and especially what's highly expressed in my tumor compared to compendium, could be a drug target. And that's the hypothesis that, of course, that's the idea. We call this outlier analysis. So this is how we found JAK1. So you can look at, so this is a particular gene, ALK, which is a known cancer gene. And so you can look at ALK expression on the x-axis. So this is the expression units. And then this is the number of samples. So most samples in the compendium express ALK in this range. But then we define an outlier range. You know, if in my case, ALK expression is here in the yellow range, then we say, okay, ALK is maybe too highly expressed. And so maybe this is an important gene for this tumor. So that's the idea. We have different flavors of this analysis. So we can compare one tumor to all cancers, or we can compare one tumor to just similar cancers, like to other brain tumors, for example. And we're kind of playing around. Well, we're investigating the best way to identify these outliers that are clinically relevant. But we can, you know, slice our compendiums, not compare to everything, but compare maybe to only relevant tumors. And we're investigating what's best. And then we can do pathway enrichment analysis. So what this means is we can take all the genes that we find to be highly expressed and put them into a map, like a functional map. So what this means for the tumor. So we have published kind of a cohort level study doing this with 4 different institutions: Stanford, UCSF, Orange County, and Vancouver, British Columbia. And so we've analyzed 181 samples in total and done this analysis. And we found that 68% of tumors had a finding like JAK1, so had an overexpression finding that could be potentially druggable. And I mentioned to you that if you just look at mutations, so you don't consider RNA, it's going to be a minority. It's going to be 10–20% of tumors that you will see a driver.
Right. So to clarify this point. So the key thing is with JAK1, it wasn't that there was a mistake in JAK1.
Right.
It's that some other mistake allowed that to be expressed very highly. So just analyzing mistakes wouldn't show that.
Exactly.
But you're going after the RNA and you're able to find out
exactly
what actually is getting turned on in ways that shouldn't be turned on.
Exactly. And of course, there is a biological question. Well, why was that JAK high? And, you know, we think there is epigenetic, you know, there is some complicated mechanism that we don't look at in the clinic. And that's an area we can study, of course, in the lab. You know, why is this JAK1 high in this tumor?
A general question. Now that we've gone through COVID, now people are able to interrupt everything. One has that kind of technology probably exploded in individual people. Yeah, is there some overlap between the viruses that are RNA that cause any problems and some of perhaps like in your research? Because there are links between viruses and diseases.
Absolutely. So we've been looking at RNA since 2015. Is definitely COVID definitely helped us be accepted a bit more. But yeah, definitely people now care about RNA more. The COVID vaccine technology, it's used in cancer. So there are trials now of cancer vaccines, right? So another application of these, we find these driver genes, so we can treat that with a small molecule. We can also make a vaccine, say that will, target all cells that have JAK1. The difficulty with that, and so that technology has been kind of exploding post-COVID in the cancer space. The difficulty that we're struggling in the pediatric field and in the adult field is targets, because JAK1, so you need a gene that's only expressed in the tumor and not expressed in any other normal cells, and that's often hard to find. Because if JAK1 is expressed very highly in this tumor, but maybe not so high, but still expressed in another tissue, then there's be a lot of toxicity to the patient. And so in leukemia space, or like blood cancer space, that's easier because even if a target is expressed in normal blood, you can transfuse the patient. So you can say, to kill the cancer, I'm going to kill off all the blood cells. And that's fine because you can bring the patient back by transfusion. But for brain or for some cell, you can't, kill the normal brain with it. And so that's been a challenge, is finding the right target, but that's an active area right now. There is like dream teams, stand up to cancer, focusing on finding good targets for vaccines and for immunotherapies as well. It's been challenging, but there is work. For example, one engineering development is maybe cancer cell, like this sarcoma cell has JAK1 high, and also has, because I showed you this very briefly, this complicated slide, but maybe it has JAK1 and then some of these other genes also. But the normal cell has JAK1, but not. So having a dual target, dual or even triple. And so that takes cell biology engineering, but that's being investigated. Kind of using Boolean logic is like you need this and this, but not this. And that's possible. It's kind of mind-blowing, but it is in development right now. So I'm very hopeful that, soon we'll see progress. Of course, it's not uniform across cancer types, unfortunately. It's like some, but we are making progress, I think. So I wanted to show you another case of how this type of analysis can help with diagnosis refinement. So this is kind of, a different example. So this is a patient from Stanford, as a 10-year-old girl with a rare tumor called immature teratoma. So this is a very interesting tumor. This is tumor they like to show in Grey's Anatomy or like medical shows, where like a young man comes in with pregnancy symptoms and then they turn out to have a germ cell tumor. So it's a tumor of germ cells, so the cells that are going to make gametes. It's like a scrambled baby, so those cells can develop into anything, and so these tumors often have like teeth, hair, like it's just a very weird tumor. But so this is the tumor that this girl had, and it metastasized to the pericardium diaphragm, which is the area around the heart. And so it was treated with surgery, and then the pericardial lesion was subjected to RNA sequencing and DNA sequencing as part of a trial and shared with us. So then what we did, and this is what we do, is we can map this patient's data onto our tumor map, right? So the tumor map is a collection of thousands of other cancers. And so it uses Google Map software. And so you're familiar with this red, maybe Google Map icon. So these red things, and this is a zoom in, and panel B shows kind of most similar cancers to our teratoma. And if you zoom in, so you may not know glioblastoma and glioma, but these are brain cancers. And so what this analysis has shown, even though this girl has a teratoma, but her teratoma is very similar to brain cancer. All of the kind of most correlated tumors are brain cancers. And the first time this was Lauren Sanders, the graduate student in the lab, we thought, okay, maybe Lauren made a mistake. We're like, we can't show this to Stanford. This does not make sense. So, but, she's investigated this, and this was the result. Basically, this teratoma, and we had other teratomas I should mention in the compendium. So it could have mapped to teratoma, but it mapped to brain cancer. And so we investigated this some more, and then we looked at markers of brain cancer. So there's already known which genes should be expressed. And so long story short here, the teratoma was similar indeed to the brain cancer. And then so we did communicate this to Stanford. And so these slides are kind of pathology view or histology. So this is a tumor put on the slide, and then a trained clinician will look under the microscope and say, this is grade, you know, whatever grade, and this is the morphology. So pathologists do that. And so when they look back, they said, yes, indeed, there are glial or brain elements in this tumor, and it was, you know, hard to spot if you don't look for it, basically. And so based on this, the diagnosis was changed to teratoma with gliomatosis peritoneae, which is a rare tumor entity, which is super rare, even more rare than teratoma, but it has been described. So this comparative analysis helped refine the diagnosis. And actually, this was a good news for the patient because this particular tumor has a good prognosis. And so that was good news. Okay, I think how much? Okay, I'm ready. I can also be done. Yeah, so okay. Yeah, so in summary, I think what I've shown you, so I've talked about cancer RNA, and I've shown that sharing data can have impact, and having these large data sets can have impact and can help refine diagnosis and identify treatment options. So we now have a lot of data that we can mine for biological insights. So questions, why is JAK1 expressed very highly? Investigating mechanisms of this high expression. So this is what we're doing in the lab. We're also applying this approach to germline diseases such as birth defects, looking at the RNA. And in that context, we're looking at the RNA to help diagnose, to help find a diagnosis. So this is the same idea. Let's look for unusual genes in this patient that doesn't have a diagnosis and see what those genes tell us. And so I wanted to thank my lab, and then I have Treehouse collaborators locally at UC Santa Cruz. We work very closely. So David Hausler, my postdoc and advisor, Josh Stewart, creator of Tumor Map, Melissa Klein, she's a scientist at Genomics Institute, Sophie Salama, professor in MCD biology, and Katrina Learned, and then our clinical collaborators, very important because we can't access patients without them, and they're very brave to be treating patients based on our ideas here. So thank you all for listening.
One question, as you look at these growing data sets, are you using artificial intelligence to start processing that data?
Absolutely. Yeah, so Josh Stewart does a lot of, he's an expert in kind of machine learning, and yeah, so we can absolutely use, so one thing we were thinking right now to do is to basically subtypes. So we have all these pediatric cancers. Can we define disease subtypes that are not based on histology? So histology, that means like where the tumor is, the brain tumor, teratoma, but based on molecular information, based on RNA. So let's define clusters that are based on RNA, but then build a program that would, when we have a new patient, classify them. So that'll be an IA that will classify them into a specific subtype, and then each subtype will be associated with a treatment. But yeah, there's a lot of data to mine here.
So epigenetics training, for instance, does anything get transferred to the seizure?
Absolutely, and so that's another reason. So thank you. So the question is about epigenetics, and so that's another reason why we look at the RNA. So a lot of these tumors have epigenetic mechanisms, but then we'll see the consequences of epigenetic deregulation in the RNA, because epigenetics will cause gene expression changes. And so we see that in the RNA. What we cannot see is the cause necessarily, by just looking at the RNA. But pediatric tumors in particular, epigenetics has a lot to do with them, and that's why they have few mutations. But they're aggressive cancers, and we talked about they're not easier to treat. So they're still super aggressive. They just have fewer mutations, and the reason for that is they have a lot of abnormalities at the level of epigenetics and then gene expression. So absolutely. And a lot of the genes we see mutated, like the gene fusion I showed you, EWSR, those mutations involve epigenetic genes like chromatin regulators, and they're very hard to target with a drug because they have so many functions, right? and such fundamental functions. They're also not at the cell surface. So yes, so the answer is yes, epigenetics has a lot to do with pediatric cancers in particular.
Even though they have some.
Yeah, I mean, that's, why, right? Like, why does it happen? Recent research kind of has identified two things. So number one, predisposition. So there is mutations in the germline that predispose to cancer that we did not necessarily know about. Also, another avenue is new mutations. So, nowadays people started having kids later in life, and especially for fathers. So if the older the father, the more chance that there'll be like a spelling mistake in the child. And so there is research showing that a fraction may be associated with that. It's not majority, but like some fraction. But the incidence of pediatric cancers are rising globally. I mean, in general, the incidence, the age of cancer onset is lowering. So there is a kind of a global phenomenon of cancers hitting us earlier and more. So there's, that like later childbearing, but also maybe something in the environment. That's one of the global challenges actually they posted in the NCI, is to figure out why the age of onset of common cancers like colorectal cancer, lung cancer is going down with generations. And then pediatric cancer is rising.
I had a couple of questions.
Yeah.
Well, one, just kind of practically what you do in your labs. So, I mean, there are people, wet labs and computational labs. You're primarily a computational lab?
So I started 2018, right before the pandemic, worked very hard to start a wet lab, and then the COVID pandemic kind of halted that a little bit. But we have a wet lab. So in the COVID pandemic, I and several members of my lab were involved and started the COVID diagnostic lab at UC Santa Cruz. And so now we have that space to apply to pediatric cancer. So anyway, my dream is to have a clinical diagnostic lab at UC Santa Cruz so that we can generate our own RNA sequencing data. So my wet lab focuses on molecular biology data generation. So we actually have an Illumina sequencer we have in the clinical lab that was bought by a donor, actually, very generous. And we're also exploring nanopore, so long read sequencing, and what that can do, to look at splicing and gene fusions and epigenetics, because we're interested in why, like the mechanism of why some of these things are highly expressed. So I do have a wet lab. It is developing.
So on the technology, so you have a person with a tumor, you get a sample.
Yeah.
So from sample to readout, how much time and how much money?
Very good question. Right now, so how much money depends on the lab. It depends if the lab does hundreds of these, right? Like if they have a high throughput facility versus they do one at a time, because one at a time is more expensive to do. At a facility, it could be like $200 to do RNA sequencing, for example.
Amazing.
Yeah, so it went down. It went down at the time. So we were involved in several of these clinical trials, and our aim was to do two-week turnaround. So that was aggressive, but we aimed to do. It's possible to do within 2 weeks, but it takes. You have to be very dedicated. Like you can't keep doing that. We could do that because low throughput. So we work with rare tumors, and we're kind of, when we get a tumor, everybody all hands on deck, and we analyze it to return the result in 2 weeks. If we were doing that every day, we need a team, shifts and,
Point out, you talked about reads, you're talking about millions of sets of reads. The computational thing is just amazing. So my other question is, seeing how you're doing this maps, you're talking about using Google Maps. So what you get is your data output is, all right, here's 20,000 different kinds of RNAs, and there's a little of this one, a lot of that one. So you cluster, looking at all the lung cancers you have, you cluster the high levels all in one patch. Yeah. And then so when you've got and you must kind of arrange your matrix kind of arbitrarily in a way, right?
So we use the approach developed by Josh Stewart, and then they have it's like so he has a physical, so principal component analysis. Maybe some of you have heard. This is one common technique to do this, is to lay out multi-dimensional data onto 2 dimensions. This is different. Like, it is similar, but first we create a 3D map where basically samples are connected by a spring, and then the correlation score that we compute kind of determines the rigidity of that spring. So the higher the correlation, the more closely the samples are, and then we allow the spring to oscillate, and then we map it to a 2D space. And so that's the approach we use, but you could do principal component analysis. You can do other types of kind of multi-dimensional, like dimensionality reduction techniques there.
Right. So you'd say if type A is high in this cancer, type B is also typically high.
Yeah, so
you
you
map them together.
Yeah, so you can ask the question, okay, we have a blob of cancers, like what defines that blob? Like what are the
Yeah.
genes that define that
Yeah.
particular grouping? And we've been doing, because that's interesting. Like, why are these cancers in one blob? And,
Right. Okay.
On the patient, the very first patient, if instead of all those aggressive treatments in the first, they had submitted you a piece of tumor tissue right away, would it have made a difference to that child at that point? I mean, even now, would it have made it if they had, instead of going through all the chemo and all the things they did, and then later metastasize, would it have helped the child to know the genomic makeup and what type, like was JAK1, would it have helped at that point?
yeah, so the question is, would it help to do this earlier? I mean, it's hard to speculate, but I know as a field, we really recognize that we should do this earlier, when the tumor is easier to control. It's sometimes hard, when we know chemo. So for example, we already have data to say that chemo cures 50% of people with this disease, or like 60%, versus this new treatment, we don't know the effect. And so it's a difficult kind of decision for a clinician, right? Like it's something that's proven to work in like X percentage. I mean, it depends on the percentage. If cases like diffuse intrinsic pontine glioma, where we know nothing works, like the conventional treatment will not work. So it's much easier to use something like this experimental upfront.
Yeah.
But if there is a treatment that, we know we have data works versus something new that conceptually sounds like it would work, but we don't know, we can't guarantee, right? So it's little kids. And so often, their parents decide for them, but this, it's challenging also at the at that level too. There is research on how that decision making takes place. We actually, in the Stanford trial, we had a nurse practitioner interviewing families about their thought process on this, and clinicians, and how do they make decisions. So that's all I can say. But I know sorry.
No. I'm just
Yeah, so the RNA expression is what you're using to identify this. How do you actually measure that? Does that mean that there's more, the abundance of RNA in a tumor cell is greater?
For that gene, yeah.
Yeah, so you're actually measuring the abundance,
Yeah.
not just the sequence.
Yeah, exactly. So that's the power of RNA sequencing. So how it works is we isolate RNA as a first step, and then we put it through the sequencing process, and then what we get back is these reads that corresponds to all the genes that are expressed, but then also from how many reads we have for each gene, we can infer how highly expressed that gene is. So you can use it to quantify.
You're doing multiple reads from the same.
Oh, absolutely. Yeah. So with this technology, and this is a technology called Illumina sequencing. This is kind of a standard technology in the field. We have to achieve, so for RNA, we aim at 40 million reads per sample. And so that would be maybe at least 10 reads for each position. Yeah.
Okay, other questions? Well, thank you very much, Elena.
Thank you.
Now, one of the things that's been going on here at UCSC is so many of the new hires are coming into more disease-related areas. So we're maintaining the basic biology, but also really getting more and more things that are applicable to medical problems.
But the basic biology is so helpful because then we can understand it, like we can, have so many colleagues that can advise me on like various genes and various things that we identify. So it's really great.
Okay, so next year we hope we'll do this again with another set of bright young investigators, of which we have.
Thank you very much.
Thank you.
Okay, thank you.
Thank you.